Welcome to this session today. My name is Mike Dolinsky. I'm with Taurus, and we're going to be talking about seed today. Although the title of this is Active Prime, it's a product that is used as a seed dressing, a nutrient packaged seed dressing. We're going to be talking primarily about how plants produce seed, what they do with that seed before they senesce, and what happens during germination, and the kind of nutrients that are involved there in the processes that take place. Now, we're looking at this canola field, and I use canola and wheat as my models for most of my presentations because canola represents the dicotyledonous plants, or the eudicots that we call them now, and wheat represents the cereals, or the monocots. So you can basically take this and use it for the germination and a senescence of any of those two crops, the monocots and the dicots. I use this slide to focus on the field and the variability. There's two issues for me around seed. Number one is the mother plant who is producing that seed, and number two is the germination of that seed when you, the farmer, put that seed in the ground. During the mother plant life, for example, in this case, we have a lot of elevation issues here. You can probably accept that plants up in the hilltop are going to be in a different environment than plants down here and plants down here and a plant a foot away from each other. The soil is tremendously variable. Some of that is related to agronomic practices, erosion, the actual formation of those soils, the organic levels, the pH levels that have been changing as we've moved into sort of zero till and we were getting a stratification of nutrients in those upper surfaces. So there's a lot of issues going on in terms of the plant uh, trying to access those nutrients, never mind the heat, the cold, the wet, the dry, all those kind of factors. So the mother plant is always working hard to try and produce a really viable seed because that's the objective of all our crops is to produce seed, and we harvest those, and that's what farming's all about. So let's start by looking at wheat. What actually happens as the plant is maturing and during that life of the plant in the field? A couple of things we need to look at is in wheat is this group of seeds called florets. Each one of these is a floret is a seed. Is called a spikelet, and it sits on a node like this that runs up the center of the spike. So when we look here, you can see that uh, these two seeds are about the same size. They were pollinated probably a day apart, and then this one maybe a day or two later, and so on. And you can see that right here, we can see the stigma still sitting there from pollination. These two seeds will be about the same size. This one will be a little bit smaller, and this will be the smallest. And you can see that each one of them is sitting, you can see it best here, sitting on a little stem, so to speak, and being fed through this node. So as the plant is picking up nutrients from the soil and water from the soil and mixing in with carbon and other things from photosynthesis, it has to feed it to each one of these seeds. And as the spike uh, continues to get pollinated, as it moves further and further away from the center, we get fewer and fewer spikelets and fewer seeds. And here just shows you that this is early pollination. As pollination moves on, we eventually get them all pollinated. And then the plant, after it's pollinated, it kicks out these anthers which produce the pollen. And that's how you know it's been pollinated. This one was pollinated probably, 24, 36 hours ago. And this one was just more recently because you can see it still is yellow and it still has a lot of nutrient left in it. Now it's usually the first flower, and you can see here in canola the first flower in this group of florets. And even in here in soybeans, the first flowers get access to the most nutrient because as the plant is approaching reproductive states, it starts to change its habits and makes the seed the sink for its nutrients. So it'll start extracting and cutting off supply of nutrients to the roots and start extracting nutrients from older leaves to supply those nutrients prior to its death, which is programmed genetically, uh, to mature the seed. So here's a final look at it. You can see here's the spike and here's the tiller, and this was done a few years ago when we had bad drought on. You can see these two seeds are, as indicated early on, are about the same size and change. As it matures, the plant loosens up the parts around the seed so the combine can basically pull it in and separate it from the spike. And you can see here there's fours, 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 3, 4, and then it goes to twos in both directions. In the tiller, we basically get twos, maybe some threes and ones. So these seeds here have the most nutrient generally, as far as I'm concerned, because as time goes on, the plant is running out of nutrients as everything is maturing. Now, the real neat thing, though, is that as the plant is maturing before it actually senesces, the plant lays down messenger RNA and mitochondria in the seed during this whole process. The messenger RNA is laid down there so that when the seed is going to germinate, it has codes there within the messenger RNA to reduce the abscisic acid, which puts the seed into dormancy, and to produce gibberellins, which stimulates growth in that seed. If it didn't do that, it would have difficulty germinating as quickly as it does. So now we're going to move on to what happens when we put a seed into the soil. We assume that there's going to be variability in the seed and that the seed is still going to germinate and it's going to be very aggressive and vigorous, and most of them do. But in fact, there is tremendous differential in terms of nutrient content of many of those seeds, and they're competing with each other. So let's take a look now at germination, what happens. We'll use wheat first. I took this picture years ago, and ever since that, I've been sort of fascinated by how seeds germinate. But let's take a look at what's going on. We have a seed coat here. We have the endosperm, which is primarily starches mixed in with proteins. There's amylose and amylopectins are the two basic kinds of starches, storage starches that are used in the seed. And it's interlaced with proteins, which gives us our 13, 14, 15, 16% protein in wheat is tied up primarily in this endosperm. The two living parts of the seed are the embryo and this little fine red layer here called the aleuron. These cells here are important in the germination process, and I'll show you that in a little while. The embryo, though, we have this membrane here, which is the cotyledon in a monocotyledon, and it's called the scutellum, and it's a membrane that transmits nutrients from the seed as it's breaking it down to the embryo to produce the coleoptile, which protects the seed, or the first true leaf going forward, and the first roots that come out called the radicle. And you can see that the actual embryo sits right on the scutellum, which is a phenomenal thing because now we have the complete mechanism to feed the new developing tissue. If we look at the nutrient content and look at what's in the embryo, the endosperm, and the seed coat, we can see that there are some interesting things that go on. We see that sulfur is mostly in the endosperm, which is fine. Sulfur is key in terms of production of amino acids with nitrogen. But more importantly, let's look at phosphorus. Phosphorus is high in the embryo and in the endosperm. Zinc is really high in both of those. Manganese is high. Boron is high. The mother plant puts the proper nutrients where it's needed to support the activity of the cells upon germination. Zinc is involved in a lot of enzymes. So is manganese. Phosphorus is there for ATP production in the mitochondria. And remember I said the mother plant puts mitochondria into the seed because the seed can't produce mitochondria unless it's got the codes for it and some mitochondria there, because mitochondria are related to bacteria, so they multiply like bacteria very, very quickly. And you'll see that in the next couple of slides that are important. So we can see that these what are called transitional metals, iron, manganese, copper, zinc, are key for dealing with our stresses in the plant called reactive oxygen species, which occur when a plant or an animal is under stress. These occur, and these are some of the key nutrients, elements that are involved in scavenging those and protecting the system. And when we look at active prime, you see that was the title of the presentation, you can see what's in it. Nitrogen, phosphorus, potassium, a lot of P, nitrogen for making those cells in DNA, boron, iron, manganese, zinc for all those catalyzed enzyme reactions, citric acid, and a chelating agent. So that's kind of what active prime is designed for, is to support the activity of that seed and to try and balance the nutrient levels that seed needs to be really vigorous. If we take a look at the location of this, as I said, the live part is the germ, which is right here, the embryo, and the aileron. So you can see the manganese is located there, the iron, the zinc. You can see how the actual embryo is loaded with zinc, and so is the aileron. As I said, tremendous amount of zinc used in plants. Now, one thing I didn't mention is that on a global basis, zinc deficiency is becoming really common, just like boron deficiency is. So in the prairies of Canada, I've been noticing more and more zinc deficiency, and did some work on wheat and found that a large percentage of wheat kernels were low in zinc. Copper is another one of those transitional metals, and it works with zinc to scavenge those reactive oxygen species, which will be in one of my other presentations. So here we have that seed again, and you've seen that, and you can see here how the endosperm is being broken down and released. Now this occurs, as I said, in the embryo where we have the abscisic acid broken down and gibberellins produced, and that activity leads to the production of alpha amylase in the aileron, which is also the life part, and that starts the breakdown of starches, the amylose and amylopectins. Secondarily, it starts producing proteases because we want to break down those proteins as well. And there are always lipases as well. There isn't much oil or fat in a seed except in the embryo here, but it'll break down the oils as well. And all of that has to be liquefied, be movable, and water is the key to that process. That amylase converts that starch to glucose or sugar so that it can then be moved through the scutellum right here into the growing shoot and the growing root. Phenomenal. It's actually a miracle of life. Now, here's where we take a close look at what actually happens. First thing that happens with a seed in first about 18 to 24 hours is absorption of water, in wheat especially, and respiration starts up. So we get the solutes being broken down into a liquid movable format. Now, here's the really interesting thing. Look what happens next. We have to repair the mitochondria because that's the source of energy in the seed to get it going. And as I said, the mother plant has laid down mitochondria in both the aileron and in the embryo. It has to be repaired because in the dry down process, the mitochondria is damaged. So it has to repair it first. And then we get protein synthesis being developed by the messenger RNA that was left behind by the mother plant to help the seedling or the seed get going. We get protein synthesized. We have to have the DNA repaired. And then after we get that water into the plant, and the plant, or the seed. The seed has to take in about 40% of its weight in water in order to get all of this rolling. Then we get everything fixed up and off she goes. The cells start dividing, the DNA is synthesized, the radicle elongates, and all those storage reserves are used post germination to grow the seedling and get it up to the soil surface. During this process as well, the reactive oxygen species are key, and the plant is dealing with all of those micronutrients and nutrients that mother plant had put into the seed. So here's what it actually looks like. Here's the coleoptile coming up, which protects the first true leaf hitting for the surface. Here's the radicle, which becomes the first root, and then these are the seminal roots on wheat. This area here is kind of a spongy area with a lot of DNA and a lot of cellular activity that's supporting both the growth of the coleoptile and growth of the radicle. And here you can see that this kernel is split in half. You can see that here's the first true leaf coming up with the coleoptile, which protects the tip of the first true leaf. And then we have the radicle hitting down into the soil. And you can see that tissue I was talking about right around here that's protecting both of those. The coleoptile hits the surface, the tip opens up, and out comes the first true leaf. The leaves are basically not yellowish or whitish because the chloroplasts in there that are photosynthetic don't turn green until they're exposed to light. And up until the first true leaf is photosynthesizing, the growth of both the first true leaf and the root is supported by the nutrients that have been stored in the seed. So that's the story on wheat. Now here's canola, and it's a little bit different. Uh, and here's a bunch of different canola seeds, and I want to point this one. Let's look at this one right here. For example, see this hole and see this structure here? Maybe this one shows it a little better, or this one here, you can see it here. Right here, you can see it as well. All seeds are the same, basically. This hole is called the micropyle, and this structure here is the actual radicle or the root. As I said, the mother plant has always already developed the sort of primordia for the root, and this hole is the micropyle is where the pollen tube enters the embryo to release the pollen or the male genetics into the seed. And that's the only entry there is into that seed because canola being an oil seed, the coating is oily. So this is where the water enters, and it's right basically at the tip of where the root is going to be. So the water enters there, and this is the first part that really gets going. And when that breaks through the cuticle or the coating of the seed, germination is complete. Here's some work done in Germany that's phenomenal. This shows the micropyle just starting to get in moisture. You can see the moisture is entering, and there's a space around the cuticle or the seed coat that the water can spread around the root, and you can see it just gradually spreads until we actually have the emergence of the radicle. Continues to spread and continues to spread. Now, the plant has to mobilize the nutrients in the cotyledons here, and you should know that there is an aileron in the canola seed. It's a small one, but the seed needs some starch, some carbon to start building molecules. So that is used initially and produces some of the lipases that can break down the oils, and then that is in fact used to feed the growth of the shoot and the root. Now, this diagram shows you that here's the radicle, but we have to have two arms because there are two cotyledons to feed that radicle, and it forms this nice network of, I assume it's xylem and phloem, but I'm not 100% sure. It looks like a venous system and almost like a lung in a human being. But you can see that they put these network of passages that hook to the radicle and feed that radicle so we can get that root out and get it rolling. Let's take a look at that and what it looks like. So here's your micropyle. As I said, the tip of the radicle will always emerge, crack it right there. So If you're thinking about it, just think about this. You put this seed in the ground. If that micropile is facing the surface and it's not able, and you're not sort of packing your seed row, the plant has trouble getting enough moisture in order to cause germination. And simply, if it's just facing downwards into the soil, and moisture will generally move from the soil up into the seed row because it's going to be evaporating, it's going to be pulling it up, you're going to get that seed germinating earlier. It's just that simple. So here's the way it's going. We get germination taking place now. The radicle continues to grow and the coleoptile is being pulled out of the seed because it's already there. The mother plant has got those coleoptiles prematurely formed. You can see that root hairs start developing very quickly. So for both wheat and canola, the seed can take in the nutrients from something like Active Prime during the imbibing process and also during early root development, where those root hairs can take any solubilized uh, elements, nutrients from that seed dressing early. And when you're putting seed in the cold ground, things like phosphorus are really key for the development of those mitochondria and the production of energy. This is the beauty of putting on a nutrient seed package. I'm a firm believer that eventually we'll realize that there's variability in the seed and we'll have a seed dressing on every seed going in the ground. The shoot is pulled, or the cotyledons are pulled up through the soil surface, and you can see here's cotyledon 1, 2. And then once they reach the surface, they turn green, start photosynthesizing, and your plant is off to the races. I hope that was both interesting and educational, and you learned something about seed. I find it quite fascinating, and I wish you the best of luck going forward. And I hope you learned something. And always remember, the plant knows what to do. All you got to do is figure out how to help it. Thanks a lot.